Thank you, Sarah, for the introduction and FDA team to invite me to present uh, pertinent generic challenges during a clinical development of orally inhaled drugs in context to US FDA and EMEA guidelines. Considering this short time frame, in next 15 minutes, I will primarily focus on generic obstacles which are definitely not distinct and have been discussed in various regulatory and pharmacology forums, but yet we have not seen any outcomes, which is basically little discouraging. And these challenges have come from, a lot of experience and are and I believe these are the primary reasons which discourages the global generics to venture into this territory of drug development, especially of orally inhaled products. This is my disclaimer, a fairly wordy slides to cut the long story short. The opinion expressed herein are solely those of mine and do not represent statements or opinion of Sandoz or Novartis Pharmaceutical. And this presentation is based on the published data. Coming to contents, this presentation will primarily include concoction of US FDA EMA guidelines comparison in context to pharmacokinetics and pharmacodynamic recommendations, common and distinctive challenges, path forward, and conclusion. In this slide, I essentially demonstrate high-level differences between US FDA generic BE approach versus the EMA. US FDA demands weight of evidence approach, which essentially means demonstrate BE in vitro, demonstrate BE in PK, and demonstrate BE in a clinical endpoint study. While EMA gives us opportunity at the in vitro stage, if not the PK stage, if not then PD stage before the application gets rejected. These differences, although they look academically intriguing, but it clearly indicates that there is no crosstalk or leverage of data mechanisms between the US FDA and EMA in context to inhaled drugs. Generics have to develop entirely a contrasting strategies for two different agencies for essentially the same innovator. Intriguingly, innovator didn't do that and leveraged the same in vitro and in vivo data for all the agencies globally. In the next slide, I will definitely go into the nuances of PK, batch selections, and clinical endpoints differences. However, there is definitely a need for harmonization amongst the guidelines uh, to make the life of generic easier. Coming to the PK differences, the basic differences in PK guidelines is that the FDA recommends PK essentially to demonstrate safety, while EMA differentiates pulmonary absorption for efficacy with interventions like charcoal block to block absorptions from GIT and safety studies, which are conventional PK studies. FDA recommends BE in all available strengths for product registration, while EMA usually accepts studies in one strength if in vitro proportionality is established. For PMDI, FDA doesn't recommend a separate spacer study while EMA does. FDA doesn't recommend pediatric studies, but EMA may recommend studies for children for some products which are essentially used in children less than 8 years old. Dose selection for the lower strength in accordance to FDA guidelines are minimum inhalation that is sufficient to characterize PK profile with highly sensitive bioanalytical methods, while EMA usually recommends clinically recommended dose which are already there in the product specific guidelines. Equivalence criteria is similar for both the agencies. However, EMA also recommends an additional Tmax similarity. For test and reference batch selection, EMA recommends a target batch, which is usually 15% plus or minus of in vitro medium, primarily with a belief that in vitro finding translates into in vivo finding, while FDA recommends a random batch approach, which is usually a nightmare for the drug development. In this slide, essentially discuss batch-to-batch variability, a primarily deterrent in generic development, common irrespective of agency guidelines. A common in vitro paradigm starts with exploratory PK studies to essentially identify critical in vitro developmental attributes which predict PK outcomes. A target reference batch is picked to develop a test in similar lines. During course of development, RLD may change, get old or expired. So during the close to pivotal PK studies, we essentially don't have a reference and a search begins to find another reference to go into PK, which requires in vitro testing of various new reference product to identify near similar in vitro specification to test and the older reference product. So how do minimal in vitro differences translate into PK differences? Here I present a published PK data on fluticasone salmeterol combination of innovator Adware, which was essentially a Sandoz data. This study, which was primarily designed to evaluate batch-to-batch variability and inter-batch residual. With this data, we understand that despite insignificant in vitro differences, there is a potential of PK bioinequivalencies in reference product, indicating a risk of failing study if RLD changes in the course of development. These PK bioinequivalencies are fundamental to inhaled product development, primarily due to low dose, low systemic bioavailability, including additional complexity of deposition trajectories in the lungs. Unfortunately, this issue has been prevailing for decades and discussed in various forums, but no guidelines have been able to address this issue. Further adding to the body of evidence to highlight the importance of batch-to-batch bioinequivalencies in PK is this statistical experiment conducted by IPAC that includes 64 subject example study in which comparisons between multiple batch and single batch approaches were made in the context of single harmonized design of two-period single group randomized crossover study. The probability of bioequivalence versus true test by reference PK ratios were plotted, and the results indicate that mere 10% between batch variability has potential to reduce the study power from 100% to 70% and inflate alpha error from 5% to 30%, suggesting that conventional single batch approach designs are not optimal study designs for inhalation products. However, in the same study, these errors were significantly negated by adding more number of batches. These results mandate more research on multiple batch designs and statistics and also probability of considering widening of equivalence limits for products demonstrating significant batch to batch variability. In the pharmacodynamic comparison, the FDA is discrete with the product-specific guidelines, while EMA is more generalized in its approach. However, the generic industry stops and inducts submissions with PK studies and generally do not venture into PD for EMA submissions, essentially due to significant costs involved in conducting these studies. FDA recommends clinical endpoint studies with lowest recommended dose to enhance the sensitivity to identify differences between test and reference. Also, along with demonstration of therapeutic equivalence, both the test and reference also have to demonstrate superiority versus placebo as their sensitivity assessment. Clinical studies for steroid-containing inhalers are usually conducted in asthma as a 4 to 8 weeks treatment study in a parallel design with spirometry parameters such as FEV1 as an endpoint, while only bronchodilator, especially the long-acting bronchodilators, are evaluated either in asthma or COPD in a crossover single-dose design. For short-acting bronchodilator, FDA recommends relative potency study using Emax model with bronchoprovocation endpoints. On the other hand, EMA recommends drug potency study designs irrespective of drug products and are open to more endpoints such as exhaled NO and sputum eosinophil. Uh, a basic fundamental issue with clinical endpoint study in a parallel design and with asthma is a large sample size. In a recent publication by FDA, which compared all the FDA submitted data, identified a variability in trough FEV1 as high as 128.6 for active product and 580 for placebo. Using this variability, a sample size of test and reference was estimated to nearly 1,400 subjects, uh, demonstrating a power of 80%, which you can see over here, plus another 100 subjects from the placebo arm. This is depicted in this red line. This variability is primarily driven primarily an outcome of inherent phenotypical and endotypical drug response and the heterogeneous behavior of asthma in terms of exacerbation rates, and also a low dose steroid response with in a wide physiological severity inclusion criteria as recommended in the guidelines. So can we reduce the high sample size studies? A basic answer is yes, if we could conduct adequately designed dose response studies or relative drug potency studies. The major challenges is that spirometry, the bronchoprovocation, FeNO, and sputum eosinophil had not been able to demonstrate any significant dose response for ICS, LABA, and LABA-LAMA combinations. And this is a data from the dose ranging studies from fluticasone furoate, which is a novel steroid. And two doses which are recommended are 100 and 200 microgram available in the market. You could see that FEV1 cannot demonstrate any significant dose association between 100 and 200. When I take this data together, uh, uh, what I see is that a 33% increase in dose would require to induce a 10% increase in response. So clearly indicating spirometry is not the answer. Uh, Other parameters such as airway resistance have recently emerged as probably promising parameters in management of lung diseases. uh, it definitely has been able to distinguish dose response, especially with the short-acting short-acting bronchodilators, and also distinguish central and peripheral drug response. Airway conductance is another parameters which needs consideration and further studies. So another important area which needs further research, which has been around for a long time, is can FDA follow the EMA way of using PK metrics to establish efficacy? Now, if we consider that API is same in its action, then elements which could be a performance measure for bioequivalence are dose available in the lung, the residence time, and the deposition geography. This is discussed, has been discussed in various forums, but I think there's still some area which needs further research. The GI, if I take PK studies and compare it with the clinical endpoint studies, the GI block PK studies essentially indicate the pulmonary dose. The metrics such as Tmax and the dissolution rates could predict the residence time, and which will definitely need a little more data. And PK studies with different APSD sizes, maybe creating homozygous particles, may be able to predict important parameters which could distinguish the peripheral and central deposition. So this area definitely needs some consideration and further research as well. So coming to conclusions, there is no strategic or scientific relationship between the FDA and EMA submission strategies, implying rather independent development programs that add to enormous cost burden in the drug development. There's an urgent need to explore novel PK approaches such as multi-batch approach, research designs and statistics for products with high inter-batch variability, as well as maybe considering the RSAB strategy and expanding equivalence limits for high inter-batch variability. Clinical trials with high sample size provide expensive problems for generic medicine makers and are significant deterrent to generic enterprises. There is also need to create innovative clinical metrics as well as study PK investigations as a method of replacing therapeutic bioequivalence, particularly its ability to differentiate against depositions in the lung areas. These are my acknowledgement. Essentially, the whole Sandoz clinical development team. Thank you very much, and I'll be available during the panel discussion to take any questions.